Hello everyone, welcome back to my channel Bunny's Biology. Today we are exploring one of the most beautifully coordinated gene regulation system in biology, that is lacZ system. But before we understand how this Operon works, we need to understand a bigger foundation what an Operon actually is. So Operon is a group of genes in prokaryotes that are controlled together under one promoter and transcribed as one polycistronic mRNA. So, Operon is a group of genes in prokaryotes that are controlled together under one promoter and transcribed as one polycistronic mRNA. So, in simple term, one promoter, one switch, multiple genes turn on or off together. So, what is polycistronic? Polycistronic means single mRNA. So, here is the prokaryotic DNA. This is the single mRNA that contain multiple cistron. So, one mRNA that translate many protein, that is called polycistronic mRNA. So, Operon is a group of gene that is controlled by one promoter and transcribed as one polycistronic mRNA that translate many protein, that is called Operon. So, in simple term, one mRNA, many protein, and this one mRNA is controlled by one promoter. So, one promoter control many protein. So, this is called Operon system. So, what is the component of Operon? So, the key component of an Operon is promoter, that is denoted by P. It is a DNA sequence where RNA polymerase can bind and to initiate transcription, and that is located upstream of the structural gene. Next is the operator, that is denoted by O. It is a regulatory DNA sequence that is positioned between promoter and structural gene, where regulatory protein, that is repressor, can bind and act as an on and off switch. Next is the structural gene, that is the actual protein coding gene that encode protein and that is required for a specific metabolic pathway and transcribed together as one polycistronic mRNA. Next is the regulator gene, that is denoted by R, that is located elsewhere in the genome and that produces the regulatory protein, that is repressor or activator, and that control the expression of other gene. But this regulator gene has its own promoter where the RNA polymerase can bind and to initiate transcription of regulator gene's mRNA. So, in bacterial cell, one of the key Operon system is the lacZ. So, lacZ is a setup where several genes operate under one promoter. So, in 1961, Francis Jacob and Jacques Monod to explain how E. coli regulates the genes needed for lactose utilization, and this regulatory mechanism known as the lacZ. So, lacZ means controls the metabolism of lactose. So, let's explore how this elegant system works. So, lacZ is a inducible system because first it is normally off, okay, and it become on when the lactose is present, and lacZ also called negative control because of the repressor protein keeps the Operon off. So, here repressor protein keeps the Operon off. So, first lacZ is off and it's become on when the lactose is available in environment, and it also called negative control because repressor protein that first keeps the Operon off. So, that's why lacZ is a negatively controlled inducible system. So, lacZ is a negatively controlled inducible system. So, let's explore how this elegant system works. So, to understand how beautifully this system works, let's break down the architecture of the lacZ. So, it contains three structural genes, lacZ gene, lacY gene and lacA gene. So, lacZ gene that encodes the enzyme beta galactosidase that cleaves the sugar lactose into glucose and galactose, both of which are used by the cell as energy source. And paradoxically, lactose also converted by the beta galactoside enzyme into allolactose. lactose into allolactose. And lacY gene that encodes the lactose permease, a protein that insert into the cell membrane. So this is the E. coli cell. In the cell membrane, permease protein is present, and this permease help the lactose transport into the cell. So lactose transport into the cell by the permease protein. And next is the lacY. lacY gene encodes the thiogalactoside transacetylase. So this thiogalactoside transacetylase that remove the toxic thiogalactosides. So this is the cell membrane, and permease protein that brings the lactose into the cell, but also accidentally brings the toxic thiogalactosides. This is toxic for the E. coli cell. But this thiogalactoside transacetylase add an acetyl group to the toxic molecule and neutralize them. So inactivate them. So thiogalactosides is inactivated by the transacetylase. So these three genes are expressed at high level when lactose is available. So these all are beta-galactosidase, lactose permease and thiogalactoside transacetylase. These all are lactose digestive enzyme. When the lactose is available in the environment, the Operon is activated and produce them. And here two regulatory protein are involved. One is activator, that also called CAF, Catabolite activator protein, and another is repressor, that is called repressor. And this repressor is encoded by lacI gene, which is located near the lacZ, but transcribed from its own promoter. This is the promoter site, and this is the promoter of the lacI gene, where the RNA polymerase is bind. So this is the lacP, that is called lac promoter site, where the RNA polymerase is bind, and this is the lacO gene. This is called operator site, where the repressor protein is bind, and this repressor protein is transcribed from the lacI gene, and this lacI gene is constitutively expressed, and this lacI gene express the lac repressor, and this lac repressor is bind in the lac promoter site, and this is the cap site, where the activator cap is bind, and both cap and lac repressor are DNA binding protein, and each bind to a specific site on DNA at or on near the lac promoter. So cap bind to the cap site and lac repressor bind to the operator site. So each of these regulatory protein respond to one environmental signal and communicates it to the lac gene. So cap mediates the effect of glucose, whereas lac repressor mediates the lactose signal. So the site bound by the lac repressor is called lac operator. So lacY gene express lac repressor. So lac repressor bind in the lac operator site. But in E. coli has three operator sequence. One is lacO1, another is lacO2, other is lacO3. This lacO1 is the primary operator, and it is the strong binding site of the lac repressor. And lacO2 that is present in the downstream of the primary operator, and lacO3 that is present in the upstream of the primary operator. And this lac operator is 21 base pair sequence. So this is the lac operator sequence. This is the 21 base pair sequence, and arranged with perfect two-fold symmetry, and also arranged as an inverted repeats. This is the two-fold symmetry and inverted sequence present, that is AATT-GT-GA. That's like AATT-GT-TA. And each half is recognized by one subunit of lac repressor. So this half is recognized by one subunit of lac repressor, and this half is recognized by one subunit of lac repressor, and forming a functional repressor dimer on the operator. So this is the operator sequence where the lac repressor is bind, and each subunit of the lac repressor is bind in the one half of the operator site. So the lac repressor bound to the operator site as a dimer form. Because of the each half is recognized by one subunit of lac repressor and another half is recognized by one subunit of lac repressor. So lac operator is recognized by two subunit of lac repressor. So the lac operator is overlap the promoter site. So this is the operator site and this is the promoter site, and this promoter site is overlapped by the lac operator site. So if the repressor bound to the operator site, physically prevent the RNA polymerase from binding to the promoter site and prevent the initiating RNA synthesis. So this is the lac promoter site where the RNA polymerase bind, but the RNA polymerase bind to the lac promoter weakly. This is because the sequence of the -35 region of the lac promoter is not optimal for its binding. So normally RNA polymerase recognize two key sequence. One is -35 region, that is TTGACA. Another is -10 region, that is TATAAT. And this sequence is called strong promoter sequence that match the consensus sequence. But in lac promoter, where the -35 region is TTTACA instead of TTGACA. So -35 region is poorly matched and also lack UP element sequence, that is upstream of the promoter, that enhance the RNA polymerase binding. So lac promoter lacks an UP element and also poorly matched -35 region. So that's why RNA polymerase weakly bound to the lac promoter site. And that's the reason transcription level also low. This is called basal level transcription. So let's see the lac operon behaves with and without lactose. So lac repressor is continuously express and bind to the operator site. So lacY gene continuously express lac repressor and that bind in the lac operator site. And that means normally lac operon is off if the lactose is absent. So lacI gene express lac repressor and this lac repressor bind as a tetrameric form and looping the DNA by binding O1 and O3 because one promoter bind two lac repressor, that is from the dimer, and two operator that bind and form a tetrameric structure. And this looping strengthens repression and physically block RNA polymerase. So if the lactose is absent, the cell refuses to waste energy making enzyme for an lactose. If the lactose is present in environment, that lactose is entered into the cell by permease. So this is the cell membrane. So lactose is entered by the permease and some lactose converted to allolactose. This is the lactose. This lactose is converted into glucose and galactose by the beta galactosidase enzyme and also converted allolactose. So this allolactose act as a inducer and that bind with the repressor protein. This is the allolactose that bind with the repressor. So if the allolactose bind with the repressor, repressor cannot bind in the operator site. So the RNA polymerase can bind in the promoter site and then the operon is on. So if the lactose is absent, then the repressor bind in the operator site, then the RNA polymerase cannot bind with the promoter site. So the operon is off. So normally operon is off if the lactose is absent because of the lacI continuously express the lac repressor. So if the lactose is present, then lactose converted into allolactose. This allolactose act as an inducer and this inducer bind in the repressor protein and repressor protein changes its shape and follow from the lacO, that is operator site, and cannot bind in the operator site. So RNA polymerase can bind in the promoter site and then the transcription is on, then the operon is on. But here you will question if the permease and beta galactoside are needed to bring lactose in and converted it to allolactose. So here lactose is inserted into the cell by the permease and this lactose is converted into allolactose by the beta galactosidase. So how do permease and beta galactosidase exist before the operon is turned on? Because this enzyme will be synthesized when the operon is fully turned on. When the operon is turned on, then the lacZ, lacY and lacA gene are transcribed. But how can small amount of this enzyme are already exist in the cell? Because the lac operon is never 100% off even when the repressor is bound. So if the repressor is bound, lac operon is never 100% off. The expression of lacZ gene is leaky. Okay, leaky expression is happened even when they are repressed. So a few transcripts are still produced because RNA polymerase occasionally succeeds in the binding the promoter despite the presence of the repressor. So the basal leakinase generates a small amount of beta galactosidase and permeates in the absence of lactose, and ensuring that some enzyme is already available to entering the lactose into the cell and convert the incoming lactose into allolactose. So here allolactose act as a direct inducer that bind to the lac repressor and induces a conformational change in the lac repressor protein. Once the allolactose has altered the shape of the repressor, the protein no longer bind to the DNA. So the lac gene are no longer repressed. So in the absence of allolactose, the repressor is present in the form that bind its site on DNA and keep the gene switched off. So now we know the function of the repressor. And now let's explore the what is the function of the activator, that is CAF, also called catabolite activator protein, and also called CRP, that is called CAMP repressor protein. So this activator function when glucose is present and when glucose is absent. So what happened when glucose is present and glucose is absent? So the activator cap function only when it binds to its co-activator, that is CAMP, cyclic AMP. So whether cap is active or not, that's depend on glucose present and glucose absent, and CAMP is active or not, that also depend on glucose present or not. So this CAMP is active when the glucose is absent. How? So this glucose indirectly inactive the adenyl cyclase. So this adenyl cyclase that convert the ATP into CAMP. So if the glucose is present, then glucose indirectly inactivate the adenyl cyclase. Then adenyl cyclase cannot convert ATP into CAMP, and CAF function when the cap is bind to the CAMP. So if the CAMP is low, then CAMP cannot bind with the cap protein. Then the cap is inactive. Then the cap is not bind in the cap site. So if the glucose is present. So if the glucose is absent, then adenyl cyclase is active, that is cannot blocked by the glucose. So then ATP convert into CAMP, then CAMP is high, then CAMP can bind with cap, then form cap-cam complex. Then this cap-cam complex can bind with the cap site. So let's see how they work. So if the glucose is present, then glucose inactivate the adenyl cyclase. Adenyl cyclase cannot convert ATP into CAMP. Then CAMP cannot bind with the cap. Then cap cannot bind to the cap site. So without cap-cam, the lac operon poorly expressed because of the RNA polymerase bind in the promoter site weakly. So the gene expression is basal level because the poorly gene is expressed. If the glucose is absent, then glucose cannot inactivate the adenyl cyclase. Then adenyl cyclase can convert ATP to CAMP. Then this CAMP can bind with the cap. Then this cap-cam complex, this is the cap and this is the CAMP. This cap and CAMP complex bind in the cap site, and this complex bind upstream of the lac promoter, that is called cap site. So this cap-cam complex act like a super activator. So this cap-cam complex helping RNA polymerase bind strongly in the promoter site. So after cap-cam complex bind in the cap binding site, this cap is recognized by the CDD tail of RNA polymerase, and that help the RNA polymerase strongly bind in the promoter site. So without cap, RNA polymerase bind in the promoter site weakly because of some changes of consensus sequencing -35 region. But with cap binding, RNA polymerase bind in the promoter site strongly and switch the lacZ operon on, and transcription level is high. So that's why cap is also known as CRP, CAMP receptor protein. So CAMP molecule is a allosteric effect for cap when cap is complexed with CAMP, that's why called CRP. So now let's look at how the lacZ operon behaves under different environmental condition in E. coli. if the glucose is present and lactose is absent, then what will happen? So if the glucose is present, that means glucose inactivate the adenyl cyclase. Adenyl cyclase cannot convert ATP to CAMP. So then CAMP cannot bind with CAF. Then CAF cannot bind in the CAP site. So low CAMP, so CAP cannot complex with CAP. So no CAP-CAM complex means no activation of transcription. So here is no activation of transcription, and also lactose absent. So lacZ that synthesize lac repressor. So lactose absent means allolactose absent. So allolactose that act as inducer that block the lac repressor. So allolactose absent means allolactose cannot block the lac repressor. So lac repressor bind in the operator site in the form of tetramer. So in the absence of lactose, no allolactose, so repressor cannot bind with the inducer and repressor does not change shape. So repressor stay tightly bound to the operator. So RNA polymerase cannot bind to the promoter site, so transcription is blocked. Then operon is repressed. So in the presence of glucose and absent of lactose, the operon is completely turned off. So this condition is called catabolite repression because lactose catabolism is repressed by glucose mediated inhibition. So we also call catabolite repression. But here the gene regulation is not completely turned off because of the leaky transcription is happened. Some kind of permease and beta galactosidase protein is synthesized by the leaky transcription. So we can ignore this leaky transcription, but we cannot say the completely turned off because the lac operon is not 100% turned off. So if the glucose is present and lactose is absent, then the gene regulation is turned off or no activation of transcription. So if the glucose is absent, lactose is present, then what will happen? So lacI gene express lac repressor. So this lac repressor can bind in the lac operator site. This is the operator site, this is the operator, and this also operator. So lac repressor can bind in the operator site, but lactose is present. That means allolactose is present. So lactose converted into allolactose. So this allolactose bind in the lac repressor. So then lac repressor cannot bind in the operator site. So the lac promoter site is free. Then the RNA polymerase can bind in the lac promoter site and transcription is on. So if the lactose is present and glucose also absent. So glucose absent mean glucose cannot block the adenyl cyclase. So adenyl cyclase can convert ATP to CAMP. Then CAMP bind with the CAF and then CAP and CAM complex that bind in the CAP site. So here CAP and CAMP complex bind this site. So the RNA polymerase bind in the promoter site is strongly. Then the transcription level is high. So if the glucose is absent and lactose is present, then the lac operon is fully activated and expression at the highest level because CAP also present and allolactose also present. Both recruit RNA polymerase strongly and transcription level is high, and this is called positive regulation where activator bound increases the rate of transcription. So activator is CAP, so activator bound increases the rate of transcription. So this is also called induction. So where lac operon is induced, so that's why called induction. So if the glucose is absent and lactose is present, then the transcription level is high. So if the lactose is present and glucose also present, then what will happen? So lactose present mean lactose converted into allolactose and lacI gene express lac repressor. So allolactose bind with the lac repressor. So lac repressor cannot bind in the lac operator site. So then RNA polymerase can bind in the promoter site. So the transcription is on. So glucose also present. That means glucose inactivate the adenyl cyclase. So adenylyl cyclase cannot convert into ATP to CMP. So CMP is low. So low CMP cannot bind with the cap. So cannot form the cap-camp complex. So cap-camp complex that cannot bind in the cap site. So without cap-camp, RNA polymerase bind weakly to the promoter site, and lac promoter is a weak promoter, we all know that. So the no activation occur. So here activator is a cap. So here cap cannot bind in the cap site. So RNA polymerase cannot bind in the promoter site strongly. So without activator, RNA polymerase bind weakly. So the transcription is low level. This is called basal level transcription. So in the presence of lactose and in the presence of glucose, the lac operon is expressed but at a low level. This is called basal level transcription. So here is the picture where the glucose also present, lactose also present. So in the cap site cannot bind the cap or cap-camp complex. So RNA polymerase bind in the promoter site is weakly. So the transcription level is basal level. And if the glucose is present and lactose is absent, then the repressor is bind in the operator site, and glucose also present, that means cap cannot bind in the cap site. So the no transcription is happened. And if the glucose is absent and lactose is present, that means glucose absence means cap is bind in the cap site, and lactose also present, that means repressor is induced by the allolactose. So repressor cannot bind in the operator site. So RNA polymerase can bind in the promoter site, and then the transcription is activated level of transcription. Here activator, that is cap, activates the transcription level high. That is called activated level of transcription. So next is the, if the glucose is absent and lactose also absent, then what will happen? Glucose absent, that means glucose cannot block the adenyl cyclase, and adenyl cyclase can convert the ATP to CAMP, and this CAMP level is high, and CAMP can bind with the cap and form the cap-camp complex, and this cap-camp complex bind in the cap site. But lactose also absent. So lacI is in that express the lac repressor. So lac repressor is blocked by the allolactose, but here lactose is absent. So lactose cannot convert the allolactose. So here allolactose is absent. So allolactose cannot block the lac repressor. So lac repressor bind in the operator site. So here lac operator site is bind with the lac repressor and form a tetrameric form. So here cap is ready to activate transcription, but activation is useless because the repressor is still bind in the operator site and blocking the operator. So RNA polymerase cannot bind in the promoter site. So even though cap is active, still transcription is off. So the activator only activate RNA polymerase when the RNA polymerase binding site is free. So in the absence of glucose and absence of lactose, then the transcription is off. So in summary, lac operon is a beautifully coordinated system that allow E. coli to express lactose utilizing gene only when they are truly needed, and lactose remove the repressor, glucose control cap-camp, and together they determine whether transcription is high, low or completely off. So understanding this condition is essential for mastering inducible gene regulation. If this breakdown of the lac operon helped you understand gene regulation more clearly, make sure to like, share and subscribe to the channel for more high yield biology lecture.